Hello and welcome to Atto Brewing videos. Today I'm going to investigate if it's possible to brew a nice looking and hopefully nice tasting beer using Spirulina, a very common cyanobacteria which you can find in many food supplements. So let's dive in. Spirulina is a cyanobacteria often found at health stores and used as a food supplement. It is often confused for being an algae, which it isn't. I will not go into the details here, but I could make entire videos on that if you want. This species here usually live in fresh water and are grown and harvested, and then you get this powder. And it's many cells put together in chains, as you can see in the picture here. And that's why they're so tiny. I've seen some people complaining that it's so fine powder, why does they have to grind it so small? It's just because they are that tiny. Ever since I started to brew, I've been interested in making a beer with some kind of spirulina addition. I have found some people trying it online, but the results are very mixed. And there are a few commercial novelty beers that include spirulina in the brewing procedure. But I have mostly not given up, but put this project on ice because I haven't found any recipe that really seems to work well. Until I stumble on an academic paper where two German dudes tried to make beer of this. And because of that, I got inspired of trying to replicate something like that today. So in the scientific paper, they were adding the spirulina powder to their beer during after flame out at 40 Celsius. So here I want to try to see what different temperatures do to the spirulina powder. So I label up 5 different experiments. One where I add the spirulina powder to 10 Celsius water. Another where I use 40 as they did in the study, then 50, 60, and lastly 100 Celsius, so boiling water. And to standardize this test, I add 2 deciliters of water to each glass, and I will add 15 milliliters of spirulina powder, so a tablespoon. And according to the package, this corresponds roughly to 2 grams of powder in each here. So that's good. And that's roughly 10 grams per liter, as they had in the study. That's roughly what they had. So as you see from the footage, I add the powder to all of the glasses here, and then I boil water in my kettle, and I pour first just tap water into my mug where I measure the temperature. Then I pour it to this measuring cylinder to make sure I get 2 deciliters, and then I pour it into the glass. And once it's in the glass, then I stir it properly to make sure that the spirulina powder is not clumped together, but evenly distributed here in the water. And the thing is that this spirulina powder is millions and millions of very tiny, small cyanobacteria, small cells. And I'm quite interested in seeing what happens when I add them like this, because we have the small particles that when I mix them here, they will get mixed in the water. But when I let them sit over time, they will sink down. And whatever compounds is extracted from these cells, it will kind of be dissolved in the water. So one way of thinking about it is kind of like me adding coffee powder to to the water here instead of algae. So the powder would sink down, but some of the flavor and color from it would get released into the water. So I will come back to these samples later once everything have sedimented down and see how just the liquid without the small particles will look like. Because this is what will probably be in the beer in the end. I don't want a lot of small cyanobacteria in there. I want just to see what's released from them. So now I will let them sit and sediment down to the bottom for a while so I can observe. Yeah, so one thing worth noting, I measure the temperature here, but then I pour it first to the measuring cylinder and then into the glass. So the actual temperature that the algae here experience is probably a little bit lower than the one I measure. Maybe it's not exactly 100 or a 60, it's probably a few degrees less. But at least I standardize it here in between. So within my experiment, I think it's quite okay. The distinct colors here, they come from different pigments that they use to harvest energy from the sun. Like chlorophyll is one we know, and they actually have chlorophyll A, which is the common one, which we found in many of the plants. I will get a little bit technical here, but it actually is necessary for the rest of the video because I will be looking into these pigments and try to isolate them. So chlorophyll A is green, and it looks kind of like a leaf. It's what you would imagine any plant looks like. And then there are two more that's interesting for this video. The first one is called phycocyanin, and that comes from Greek. It means phyco means that it's an algae, which it isn't, and cyanin is cyan, so it has the color, a blue color, and found in these small, not algae. And so we have chlorophyll A, which is green. We have phycocyanin, which is cyan or blue. And then the third pigment that's interesting today is also called phyco and phycoerythine, and that means it's red. So we have three colors today: green, blue, and red. And that's all inside of these cyanobacteria. And this will, of course, affect how the beer will look in the end, because we have actually one more color to play with, and that's the color of the beer itself in which we add this, which is usually some kind of yellow. So what I'm investigating here today, or trying out, is what happens when you add this powder in different ways to beer. And of course, yeah, there are also more pigments than the three I mentioned here, but I think I'm already too technical, so we'll leave it at that. This is directly after I added all the water. Here we have the 10 Celsius, and you can see it's kind of foamy here at the top. You see a lot of particles, it's very green, but there is a bit of foam on the top surface. And now for comparison, I will bring the 40 Celsius and the 100. The 100, you see, it doesn't have this foam, and it's not so vibrant, not so green-blue, it's more just dark. And you see the foam here on the 40 Celsius. So, and here you can actually see the particles in the 100. You see there are small, tiny particles in the water. This is what will sink down in the end, and it's just kind of greenish. So I'm not sure, but one theory I have is that these, here you see it's another green color. It's more of this blue-green, this cyano color in it you can see. So one theory I have is that these foam you see at the top is actually different sugars that are dissolved. Here is the 60 Celsius one, and you see a lot less of these sugars. It still have this like blue-green, not as the 100, which is just like pale. So it still have the color, but not the foam. And here is the 10 for comparison. So 10 and 60. And you can see the foam here. So I guess the higher temperature kind of breaks this down a little bit. I think this is the 51, and it's the same. The higher you go, the less foam you get. And then in the 100 Celsius treatment, you lose a lot of this cyano blue and only get the green. And I would assume that this kind of destroys a lot of the pigments. Here you see this cyano color quite clearly. Yes. And here in the 100 Celsius, you see the green without the blue, and it's also a bit more translucent than the 60. So here we have a top view of the different treatments, and I think it's quite clear here. The more to the left you go, the more foam there is, and the more to the right, the less. And then furthest to the right, it looks different, almost green, and that's probably because the other pigments here are damaged. You don't see it so well in the video, but it's almost like see-through. The other ones are much thicker. The light doesn't really penetrate, while the one here furthest to the right is more translucent. And I really think that speaks for the high temperature here, destroying the different pigments. Okay, so now this is the same evening, but I got hold of my dive light because I wanted to clearer show you what you see here. So on top, I think there is some of the cells. So it's the chlorophyll you see here, that's very green. But you see underneath, we have a different color. So the red color here, it looks very similar to the phycoerythrin that I showed earlier. And when going higher and higher in temperature, we still see it. I think this here is the 50. And in the 60, we have the same. I don't know why there seem to be less of the cells on top here in the higher temperatures, but that seemed to be a pattern. Maybe less sugars is keeping less of them in suspension. But anyway, here we have the 100 Celsius, and here we see it's only the chlorophyll left. The phycoerythrin, the red pigment here, is destroyed, it seems like. And just for comparison, here is the 10 Celsius again, and here is a lot of pigments left. So here we have a side view, and you see not very much red color until I turn on the light, and then it's very evident. And you see here too that everyone except the 100 Celsius to the right have this red color when I shine the light on it. And you can see very faint in the very right that there is a bit of cells at the bottom. It's quite hard to see here too, a little bit of green at the very bottom here. We're going to see more of this the next day. Here you see almost all the cells here have fallen out of suspension. They have sunken to the bottom in the 100 Celsius, much quicker than the other ones, it seems like. So now we're back a bit later, where the cells have had more time to sink to the bottom. And I start off by showing here the difference between the 10, the 60 and the 100. And you see all the cells here at the bottom and how translucent the 100 Celsius really is. And we still have the foam here in the 10, which is missing in other ones. I also tried to extract some of the pigments using ethanol. I only have vodka, so that didn't work, so I will not talk more about it. Here I discovered something interesting. When you don't shine the light directly on the liquid, but at an angle as here, you quite clearly see the phycocyanine, the blue pigment here. Not when you shine it straight through, but from an angle like this, you see it quite well. You also see a faint shine of the red too. But yeah, I think that's really cool. Okay, so now it has been sitting for 24 hours and had even more time to sediment down. So what I want to do now is to see if I can filter out the cells here. Each cell is just like a few micrometers, but they can form quite large colonies. So I hope that a coffee filter here will be enough. I think the pore size is around 20, if I remember correctly, micrometers, but I might be wrong. Yeah, but what you see here is that the water is still quite green, and when I inspected it clearer with my eye afterwards here, it was not good enough. This filter didn't really remove the cells. They were still left. And I tried the same thing here for the 60 Celsius. And here you see the stratification quite clear, the green layer at the bottom and the red above it. And now the red is quite clear even without the dive light. I think it's because all the cells have sedimented down, so we only have the red and blue left in suspension here, like that's dissolved. And here we see the blue very clear, but as soon as you get more and more, the red kind of takes over. So it really depends on the light and how much liquid you have, really, which color it appears at, which I think is very cool. And you see here when I pour and spill, it's very blue, but when you have the more of it in the glass, it's very red. And especially if I turn on the dive light, then even the droplets on the bench become red. And here to the left, you kind of see the green here at the bottom. It almost looks like a candy. You see all the chlorophyll in all the cells on the bottom, and then the other pigments dissolved in the water. So my plan is to try to add a bit of this liquid here to a beer and then see how it looks and tastes. I have also tried the liquid here as it is, and it was very metally, and I really did not enjoy the flavor. I'm not a fan of the spirulina flavor to begin with, but it was too much for me. It was like drinking metal. So I hope it goes better when I add it to the beer. So now it's time to test it with the beer here. I used a lot less than what I did in the study because it became very, very blue. And I wanted something that was a little bit see-through that still kind of looked like beer. I will put the exact concentrations here on the video in text, but I took 2 deciliters of beer. This is the rice lager from our previous video. And then I added the concentrate here that I made earlier. I took 1 teaspoon, I think? No, tablespoon, and added to the beer here. And as you can see in this light, it looks very blue. And the beer is very yellow. And when added, as you can see, we get green. It looks very, very nice. Maybe good for St. Patrick's Day. I don't know. Maybe next year. So this is how it looks. And yeah, I really like the color of it. I think it looks very almost toxic. Here we go. I think you can still see a little bit of the blue in it. And when trying out the dive light, you can see a faint red shade here, but it's not at all as strong as it was before. But there is still a little bit of it left, but much less. The green is really the predominant color here, I think. I tasted this beer. It was still a lot of metal color taste in it. Even though it's much better than just the cells, it's still, at least to my taste, is way too much. So my plan now is to add this to 2 bottles of beer before I bottle it and let it sit and then see how it turns out in the end. Also in the paper, they tried to do a 60 IBU American Pale Ale that was quite sweet and bitter, and they at least claimed to have really good success with that it was much more balanced and actually tasted nice, even though it's much more spirulina in that beer than I have here. So if I don't have a huge success with the next test here, I will probably try to do something like that. But this video has been going on for way too long now, so I will have to round off for today. If you have any suggestions or ideas here, I'm very open for that. But yeah, thank you very much for following along here in adventure. We haven't really found the perfect recipe for spirulina beer yet, but the hunt is still active and ongoing. Cheers!